Okay, here's a quick rundown for the practice problems from week 8. So let's start with question 1. Question 1 went over the chromosomes in elk. So it says that elk have a total of 68 chromosomes. With the total, we know that typically elk are diploids, so that means that if we divide that by 2, that they have 34 different chromosomes, and then they are in sets of 2. So one is the maternal and one is the paternal. So now we'll start with part A. It asks how many total chromosomes the elk would have if it were actually a tetraploid, so tetra meaning 4. So instead of just the typical diploid that most of these mammals are, tetraploid would mean that we have 4 of each type of chromosome within there. So you would take 34, which was the original number of types of chromosomes, and multiply that by 4 and get 136. Okay, so moving on to part B. Now, if our elk, having the total of 68 chromosomes and being a diploid, what would happen if the elk was disomic for chromosome 5? So di is referring to the number 2. So with chromosome 5, we already have our 2 sets because the elk is a diploid. So we would follow the same layout. We would have the 34 total chromosomes multiplied by 2 since it's a diploid, and it's already disomic for chromosome 5, so we'll have a total of 68 chromosomes. Part C asks what would happen. Am I in part C? Yeah, part C asks what would happen if the elk were trisomic for chromosome 17. So now over here, instead of having the 2, we have a third one added on, making it trisomic. And that would mean that we would take 2 times 34 for the total number of chromosomes, getting 68, and then we would add an additional 1 for that third extra 17th chromosome, giving us a total of 69 chromosomes. Part D asks if the elk is monosomic for chromosome 23. That means that over here, number 23 loses one of the chromosomes, so instead of being disomic, it is now monosomic. Same thing where we take 2 times 34 since the elk is a diploid organism, and then we're going to subtract 1 from that total amount because of the monosomic 23. And we'll have a total of 67 chromosomes in the elk. And then finally for part E of question 1, if the elk has a reciprocal translocation between chromosomes 5 and 17, how many total chromosomes will it have? So the reciprocal translocation simply means that during crossing over, sections of the chromosome 5, one of them switches with chromosome 17. And back and forth, so 17 will go on to 5. In that case, even though those chromosomes are now a little more mixed up, the total number is still going to be 68 since chromosome 5 still technically has 2 chromosomes and number 17 has 2 chromosomes as well. Okay, I'm going to move on to question 3 now. Question 2 is primarily with vocabulary, so I'm going to cover 3, 4, and 5 since there's a little more steps or details to it. So question 3 is about an individual that has an inversion in one of their chromosomes. So here we see for this individual they have these 2 chromosomes. I highlighted in blue is the paternal, and red is going to represent the maternal. So the paternal chromosome is in the correct order, no inversion has taken place, but the maternal has an inversion. So I've underlined here the section in both of them that is either normal for the father, or not the father, but the paternal chromosome, and then flipped in the maternal chromosome. So now the question asks, what is going to be the outcome, the 4 products of these chromosomes after meiosis takes place? So prior to meiosis, all the chromosomes replicate. So I've shown this here just by making a little duplicate. And then the asterisk here is representing, I'm using that to show the little dot, the kind of bullet point dot that's in the middle. That's where the centromere, where the two sister chromatids attach. So you can see that on both of these here. Okay, so now it wants to know the four products that you're going to get from that if a crossing over occurs between the E and the F alleles. And just as a reminder on here, for this question, if the letters are capitalized, like in the paternal one, then it's dominant. If they're lowercase, then it's recessive. It doesn't apply too much, it just is helping you to keep track right now. Okay, so it's important to point out that this is taking place during prophase 1. That's when crossing over occurs. That's when the homologous chromosomes that have duplicated come together and form a tetrad, and then you can have that swapping of genetic material between some of the sister chromatids. I've highlighted here in orange on both the father's sister chromatid and the mother's that are adjacent to each other, the area where the crossing over is taking place, so between the E and the F alleles. Now you can see here, let me back up really quick, you can see here that right now the F lines up with the E and the E with the F, so they aren't perfectly matched. So to make this crossing over a more likely situation, I'm going to flip the maternal chromosome around, so it's basically written backwards here. But when that takes place, now we can see that there are, the letters are lining up now, so C, D, E, F, G, H, so that increases the chance of crossing over taking place, and here it's going to take place between the E and the F. Okay, so now that the paternal and the maternal chromosomes are more closely aligned, we're going to have the crossing over take place. So again, this is during prophase 1, and this genetic material is going to get swapped. So some of the maternal genetic material went over to this sister chromatid, and then the material from the paternal one went over to the maternal here. So once crossing over has completed and we move past prophase to metaphase and then to anaphase, our homologous chromosomes get split apart, and so the paternal one will go into one cell and the maternal will go to another. And then they'll go through another round of divisions for the second half of, or second phase of meiosis. And at the end of meiosis, we're going to have 4 total cells, each with a different one of the chromosomes in them. Okay, so technically these 4 products are the answer for part A, but just to make it match up perfectly with what you can get in the key, the maternal ones, I'm going to flip them back in their normal direction. So if you flip them in the opposite direction, then this will line up perfectly with what you have on the key. Okay, so part B for the question, for question 3 now asks, which of these 4 chromosomes, these final product, are expected to produce viable offspring, meaning that the chromosome in it provides enough material that represents the original so that offspring would be able to form. So in this scenario, the first one and the last one here would produce viable offspring, and it's because they have the full, basically in this context, they have all the letters. So all the different genes that are associated with that chromosome are there. And both of these ones haven't been touched at all. So the other ones will not, which then leads us to part C. So part C asks, which of the 4 chromosomes contains deletions or duplications? And that would be these middle 2. Sometimes you can have chromosomes that have deletions or duplications and you can still have a viable offspring. It's just a lot less likely, which you've gone over some of those cases in the material for week 8. And the reason why offspring with these chromosomes, with these deletions or duplications, typically aren't viable, why they don't develop, is because they are typically missing too many of the genes that are on the chromosome to be able to produce the organism. Okay, and finally moving to D. Which of the 4 chromosomes are acentric or dicentric? So centric is referring to, and these things, the little asterisks or on your paper, the bullet points, which are representing the centromere. So if it's acentric, the A typically means that it means non, or it doesn't have it. So which ones either do not have that centromere, or if they're dicentric, does it have 2 of them? And in this situation, the middle 2 again are. So this top one is acentric, does not have that centromere portion, and the lower one here is dicentric, meaning it has 2 of them. Okay, on to question 4. So question 4 is dealing with an individual that has had a reciprocal translocation take place. So in question 3, the one we just went over, that was focusing on just one type of chromosome. So for example, it could be chromosome 3 or it could be chromosome 17, and it was just dealing with the maternal and the paternal version of it. In this one, we're dealing with 2 types of chromosomes. So up here at the top, the ones that start with AB, or ones that go A, B, C, D, E, F, G, this is one type of chromosome, and we can have the maternal and the paternal version. And then the second chromosome is this RSTUV, and you can have, again, the maternal or the paternal version of it. So again, to recap, in this question, we're dealing with 2 types of chromosomes. One type up here, the A, B, C, D, E, F, G, and one right here, which is the RSTUV, and we have 2 of each because we have the maternal and the paternal of each. Okay, so prior to meiosis, replication will take place, so we're going to duplicate each of those. And now we're going to go through a few different types of translocation. So for part A, it says, suppose the translocation cross undergoes the alternate pattern of segregation in meiosis 1, and it wants to know what's the chromosome composition in the 2 daughter cells at the end of meiosis 1. So not at the end of all of meiosis, but just halfway through that first phase. So when you're doing these problems, you want to keep track of, one, what stage of meiosis do you need to account for them. So in this one, it wants to know what are the 2 daughter cells at the end of meiosis 1, and then you want to see what type of translocation cross is taking place. So in this situation, we are having an alternate pattern. Now, I like to set up, my way of solving this is to set up a little grid. It helps me. So I marked here which one, this is going to be chromosome 1, these green ones, the A, B, C, D, E, F, G, and then chromosome 2 is going to be the ones in blue with the RST. So right here, I should note, in case I haven't already, that with these chromosomes, we've had a crossing over take place. So a section of chromosome 1 has been swapped with a section of chromosome 2. So this sister chromatid has swapped some of the material, and that's where you see I tried to keep it so it's green and blue so you can see that difference there. So like I said, I like to do a little trick. I make a grid, so you can see sometimes they'll draw the chromosomes out in this weird kind of diamond shape where they line up. What I do is I make a grid. So on the right side of the grid are where the chromosome 1s are placed, and then on the left side of the grid are where you place the chromosome 2. In the top right corner, I place the normal version of chromosome 1, so the one where no crossing over has taken place with the other chromosome. And then in the bottom left corner, I put the normal chromosome 2, so they are on opposite sides of each other. That doesn't make sense. They are kitty corner to each other. Or diagonal. On the these other chromosomes that I'm going to mark with the little, I don't know what that's called, little pokey, spiky bubble speech. Okay, so those ones, chromosome 1 and chromosome 2, these represent the chromosomes where the crossing over took place, so where there's a little bit of chromosome 2 on chromosome 1 and vice versa. Okay, so now I'm going to start filling in with the actual chromosome. Chromosome 1, I place right here, and these are again in their duplicated state, and chromosome 2, and then down here for the the chromosome 1 that has a little bit of the swap material in it, I put that down there, and then up here on chromosome 2. There we go. So first we're going to figure out what happens when an alternate pattern of segregation takes place. In this situation, when the cells separate at the end of meiosis 1 to form the two daughter cells, these chromosomes will separate into one of the cells, and these chromosomes will separate into the other one. So you'll end up with two cells, one that has both chromosomes without any of the swap material, and one that has both of the chromosomes with swapped material. Okay, so for part B, now it's asking what will those two daughter cells look like if an adjacent 1 pattern of segregation takes place. So in adjacent 1, the top quadrants of the little grid that you make, those chromosomes are going to go into one of the cells, and the bottom quadrants are going to go into the second cell. So both of these cells will not have all of the material, the genetic material that they need, because while it has everything for the normal A, B, C, D, E, F, G, H, I, here on the other chromosome it has the first section of it, but we don't have X, Y, and Z. And then the same thing down here, but instead we are missing the G, H, I portion. Okay, and so for part C, now it's asking what will those two daughter cells look like with their chromosome content when an adjacent 2 pattern of segregation takes place. So in adjacent 2, the right quadrants are going to have, those chromosomes are going to go into one of the cells, and then the left quadrants, those chromosomes are going to go into the other cell. So again, you're going to have cells here where you don't contain all of the genetic material, because in this case we're missing quite a bit of it because we don't have any of the remaining part of the quote-unquote the blue chromosome, and down here we're missing a great deal of the green chromosome. Okay, finally in question 5, it wanted you to draw out what it would look like if non-disjunction took place either during meiosis 1 or meiosis 2, and what that would look like in the resulting daughter cells. Okay, so let's start with non-disjunction in meiosis 1. So our first round is, it wanted you to keep track of metaphase 1, anaphase 1, and telophase 1 in cytokinesis, and then for the second round you're going to keep track of metaphase 2, anaphase 2, and telophase 2. Now I've marked on here 2 types of chromosomes, so we're going to have the blue and the green. And instead of color coordinating the difference between the maternal and the paternal, I just put a P and an M to keep track. So right now I have the paternal for the blue on the bottom and the maternal on top for this first set of chromosomes, and then on the right with the green I have the paternal on the top and the maternal on the bottom. Though this could line up differently, I just chose it this way just because. Okay, so in metaphase 1, the chromosomes line up in the middle in the metaphyseal plate, which I think they asked you to draw that out. I didn't, but it would be lining up right across here in the cell. Then during anaphase, in meiosis 1, homologous chromosomes separate, not sister chromatids, homologous chromosomes separate. So if it went well, then one of the blue would go to the top, one to the bottom, and the same thing for the green. But since non-disjunction took place, I had both of the green homologous chromosomes, the maternal and the paternal, coming down to the bottom here. Okay, so after anaphase with telophase, you're going to pinch off and in cytokinesis, and you're going to have nuclear membrane form So I forgot to draw that in, but I did it here on the PowerPoint with a little orange. So during telophase and cytokinesis, the nuclear membrane will come back and then it will dissolve again going through the second round. So right now we have two daughter cells. Both of them have one of the homologous chromosomes of the blue, so one each, and then this bottom one here has both of the green chromosomes. Okay, so now we're moving on to meiosis 2 for this version of non-disjunction. So now we take these two daughter cells and they're going to go through another round of splitting. So our chromosomes are going to line up in the middle at the metaphyseal plates, which for this bottom one is going to be right across like this, and for this top one will be right across like that. The spindle fibers will attach to them, and then now the sister chromatids are going to split. So in anaphase 2, sister chromatids will be pulled apart to opposite ends of the cell. And then for telophase 2 in cytokinesis, those cells will then be split down the middle. A nuclear membrane will form around, and our final product in each of these four cells, these top two, will have one chromosome, and it will be one of each of the maternal blue chromosome. And then these bottom ones will have one each of the paternal chromosome, but then they're going to have one each of both the maternal and the paternal green chromosomes. So you're going to have two cells that have one chromosome each, and then two cells that have three. Okay, now we're going to see what happens when non-disjunction takes place during the second half of meiosis, during meiosis 2, instead of during meiosis 1. So we're going to start again with our initial cell, and it has two types of chromosomes, the blue and the green, and then we have the maternal and the paternal types of each of those. And this time I lined them up so both the maternals were on the top and the paternals were on bottom, but again those could line up differently. Again, they were lined up at the metaphyseal plate. When they separate during meiosis 1, homologous chromosomes separate. So the sister chromatids remain intact, but the maternal and the paternal chromosomes are pulled away from each other. In this version, in meiosis 1, everything went well, so we have in our final two daughter cells of meiosis 1, we have the one of each type of chromosome, so we have a blue and a green in each of these daughter cells. And in this case we have both paternals over here and both maternals over there. There could be, if I had lined them up differently at the beginning, we could have had a maternal and a paternal and vice versa. I put here the nuclear membrane forms again and then will again dissolve as we go into meiosis 2. Okay, so during metaphase 2 of meiosis 2, our chromosomes line up at the metaphyseal plate, right along here for this cell, and right along here. And the spindle fibers will attach on, and this time the sister chromatids are going to separate during anaphase. So they pull them apart, but this is where our non-disjunction takes place. So in this bottom cell here, I'm showing it working out just fine. So the sister chromatids split, and one will go to each end of the cell. But at this top one, the blue chromosome, those sister chromatids split just fine, but the green one didn't, and those ones remained attached. So in this one, we don't have any green chromosome being pulled at the top, but both of them are being pulled to the bottom part of this cell. So now, when these cells divide, and we have telophase and cytokinesis taking place, we will end up with 4 daughter cells. One of which will have one chromosome. It will have, in this one, it has the maternal blue chromosome. We will have 2 cells that have the standard or the normal version, so they have one of each type, one blue and one green, and both of these have one set for the paternal. And then we're going to have one cell that has 3 chromosomes in it, and in this one it has one of the blue, the maternal one, and it has 2 of the green, both of the maternal ones. So hopefully this video helped a little bit to just kind of give you a visual and walk through the steps for solving these problems.